Hello, everybody. It's Damla and Dr. Sven Vilain join us today. Could you introduce yourself, Doctor?
Yes, I'm Sven Vilain. I'm assistant professor at Istanbul Medical University. During my PhD and postdoc, I studied the function of certain genes which are involved in neuronal development and neuronal degeneration. I studied their function in the fruit fly model organism, and currently I'm studying the function of genes that are involved in regeneration, and I'm studying the function of these genes in the highly regenerative model axolotl.
So today we are here to discuss the 2022 Nobel Prize in Physiology or Medicine awarded to Svante Pääbo. So start with who is Svante Pääbo?
Svante Pääbo was born in 1955 in Stockholm in Sweden. So when he entered university in 1975, he first pursued a degree in Egyptology because he was very fascinated about Egypt. But it was too much linguistic oriented, and soon he switched to medicine. When he was studying medicine, he interrupted his education for a while to pursue a PhD in molecular genetics. And during his PhD, he realized that there are so many technologies that people are using to clone DNA, but nobody really seemed to have applied it to archaeological remains, in particular Egyptian mummies. So he wanted to try to amplify DNA from mummies. And then with the help of an Egyptology professor, he gathered some tissue from mummies, and he tried to amplify their DNA. And then he was writing his paper on the DNA that he discovered in the mummies, and this is like where his research interest started.
Okay, so why do you think Svante Pääbo win the Nobel Prize?
Well, we have always been wondering where we come from and how we are related to those human ancestors who came before us. So with his research, he could shed some lights on these questions, and with his important discoveries about the genomes of extinct hominins and human evolution. So let me clarify the term hominin a bit more, because I'm going to use it talk about hominins. So hominins is actually a term given to all the humans and all the ancestors from the humans who walked on 2 feet. And the most famous hominin is Neanderthaler, and Svante Pääbo managed to sequence its genome. On the other hand, you have hominids, which is a term given to all modern and extinct great apes, and it also includes humans. But hominins is more really humans and all the human ancestors. So comparing the genome of Neanderthal and Denisova with modern humans, he discovered that there has been some gene transfer between the extinct hominins and modern humans. So during the migration out of Africa 70,000 years ago, Homo sapiens met the now extinct hominins like Neanderthal, and there was some exchange of genetic material. So we got some gene variants that are coming from the extinct hominins, and they kind of contribute to the variation in the modern human genome, and therefore it has a lot of relevance to this day. For example, it can affect the immune system. On the other hand, the difference between the genome of these extinct hominins and the human genome can teach us something about what makes us specifically human. So which part of our human genome, Homo sapiens genome, sets us apart from the extinct hominins.
Okay, so where do you come from?
So research predicted that the anatomically modern human, Homo sapiens, first appeared in Africa around 300,000 years ago. At the same time, our closest relatives, like Neanderthals, developed outside of Africa and populated Europe and Western Asia. And this happened from around 400,000 years until 30,000 years ago. At 30,000 years ago, they went extinct. And then 70,000 years ago, a group of Homo sapiens migrated from Africa to the Middle East. And from there, they spread to the rest of the world. So Homo sapiens and Neanderthal coexisted in large parts of Eurasia for tens of thousands of years.
Well, if we coexisted, then we must have exchanged DNA. How can we know this?
So if you want to answer this question, we need to compare the genomes from modern humans and the genomes from the extinct Neanderthals. So around 2000, the entire human genome, or almost entire human genome, had been sequenced. But of course, you want to compare the human genome with the genome of extinct Neanderthals. And for that, in order to manage to do that, we need to get a genomic DNA from these Neanderthals. So if we have both sequences, we can compare both, and we can compare the genomes for modern humans with Neanderthal.
So how did he manage to sequence the Neanderthal genome?
So it was not easy, so he needed to solve some very hard technical challenges. First, over time, for example, DNA becomes chemically modified and it degrades in very short fragments. As a result, after thousands of years, you will only find very small amounts of DNA, which is very short. And second, these trace amounts of DNA are massively contaminated with DNA from bacteria, for example. I mean, you can imagine that if a Neanderthal person dies, his body starts to decompose, and you can imagine that the DNA of the bacteria and fungi that are decomposing the body will get mixed with Neanderthal DNA. And third, because there's so few DNA left, any DNA contamination from modern humans, even when it's very, very low, it will still affect the results. Therefore, it took Pabo several decades to develop the methods necessary to study the DNA from Neanderthals. So first he decided to analyze the DNA from Neanderthal mitochondria. So why did he use mitochondria? So our human cells, they have genomic DNA. So they have 2 copies of genomic DNA, which is in the nucleus. But it's not the only DNA that our cells are having. Also mitochondria, which are small organelles inside our cells, they have their own DNA, which is very small and circular DNA with very limited amount of genes. But because each cell has many mitochondria and each mitochondria has many copies of its mitochondrial DNA, therefore there's a lot of mitochondrial DNA present in tissues. And he wanted to make sure that his method, that he wanted to increase the chance of success to analyze the DNA from extinct species. That's why he used mitochondrial DNA, just because there should be much more of it. And then with his refined methods, Pebo managed to sequence a region of mitochondrial DNA from a 40,000-year-old piece of bone. And then for the first time, we actually could see a sequence from extinct relative. Also, when he checked the sequences from this mitochondrial genome from Neanderthals with modern humans and chimpanzees, he could see that the mitochondrial genome from Neanderthals is not like chimpanzees and not like humans. So it's kind of has its own DNA.
So how did he sequence the full Neanderthal genome then?
So he further improved the methods to isolate and analyze DNA from old bones. And also some new technical developments were made, which allowed to sequence DNA very highly efficiently. He also took some collaborators which had some expertise in analyzing sequencing. Basically, how I understood it is they could isolate a small DNA fragments present in Neanderthal remains. These DNA fragments, like I said before, they are also contaminated with bacterial DNA fragments. And they're also very low, so there's very small amounts of these DNA fragments. So first he needs to amplify these DNA fragments to get more of them. He could do that by growing the DNA in bacteria. Second, they needed to determine the correct DNA sequence from these fragments. So now you have these fragments, you have a lot of them, but you need to, a lot of the same, but you need to figure out what's the DNA sequence. Fortunately, new efficient methodologies allowed to do this. And in this way, he could obtain sequences from many small DNA fragments, but like I said before, these DNA fragments were still a mix of hominin, Neanderthal DNA, and contaminated DNA. So how can you distinguish which is DNA from Neanderthal, which is DNA contamination? So at the end of last century, the human genome was sequenced, and people know when they compare the human genome with the ape genome that they're very, very similar. There's a high level of similarity between both. So they could also guess that the Neanderthal genome should be very similar to both. So then he had these small DNA fragments which he amplified, and he knew the sequence from these small DNA fragments, and he has the human genome. So now he could kind of check which small DNA fragment kind of corresponds to which part in the human genome, and in this way he could puzzle the whole Neanderthal genome together. And he published this work in 2010.
Next question is, what did this sequence teach us about our genome?
So comparative analysis of the DNA sequences demonstrated that most recent common ancestor of Neanderthal and Homo sapiens lived around 800,000 years ago. And then from this ancestor, both the Homo sapiens and Neanderthal evolved each in their own way, and also accumulating a Neanderthal and Homo sapiens specific, more specific mutations. So now Pebo and his co-workers could also investigate the relationship between Neanderthals and modern day humans from different parts of the world. So what he found when they compared the DNA from Neanderthal with the DNA sequence of modern day humans from Europa and Asia, he found that approximately 1 to 4% of the Neanderthal genome is present in these genomes. If he also checks the Neanderthal genome and he compares it with the DNA sequences of Africans, he could not find this presence of a Neanderthal genome. Therefore, it seems that when humans migrated out of Africa, they encountered the Neanderthals and interbred in the places where they were coexisting, while the modern humans who remained in Africa did not encounter these Neanderthals.
Pebo also discovered a new hominin, right? Could you tell something more about this?
Well, in 2008, a 40,000-year-old fragment from a finger bone was discovered in the Denisova cave in the southern part of Siberia. This bone contained exceptionally well-preserved DNA. So interestingly, the DNA sequence was unique when he compared it to sequences from Neanderthal and present-day humans. So he was trying to figure out whether this DNA was from Neanderthal or humans, and it turned out to be not from Neanderthal and not from humans. So he discovered a previously unknown hominin, and he gave it the name Denisova because it was discovered in the Denisova cave. When he compared this sequence from contemporary humans from different parts of the world, it also showed that there had been a gene flow between Denisova and Homo sapiens. So he found, for example, that population in Melanesia and other parts of Southeast Asia, that there's individuals that carry of up to 6% of the Denisova DNA. So Pebo's discoveries have generated a new understanding of our evolutionary history. At the time when Homo sapiens migrated out of Africa, at least 2 extinct hominin populations inhabited Eurasia. You had the Neanderthals living in Western Europe, and you had the Denisovans population, mostly the eastern part of the continent. And when the Homo sapiens migrated out of Africa, they met both groups and they interbred with both groups. We also know now, for example, that Denisovans, that they kind of diverged from Neanderthals, so they kind of sub-branched from Neanderthals that evolved independently.
This research gave rise to an entire new scientific discipline called paleogenomics. What is paleogenomics, and could you tell us something about the relevance of paleogenomics?
So paleogenomics is a field of science which is based on the reconstruction and analysis of genomic information from extinct species. And the field relies very heavily first on improved methods for extraction of ancient DNA. And this ancient DNA can come from museum artifacts, from ice cores, from archaeological or paleontological sites. And second, it also relies on very efficient DNA sequencing methodologies. And based on the groundbreaking work from Sven Pebo's group, for example, he found several additional genome sequences from extinct hominins. So Pebo's discoveries has established a unique resource which is utilized extensively by the scientific community to better understand human evolution and migration. And new powerful methods for sequence analysis indicate that also other ancient hominins may have mixed with Homo sapiens in Africa. However, we cannot find these other ancient hominins DNA remains because in the tropical climates the DNA, very old DNA degrades very, very fast.
Okay, so what are the consequences of having Neanderthal or Denisovan genes in our genome?
So thanks to Sven Pebo's discovery, we now understand that archaic or very old gene sequences from our extinct relatives are present in our genome, and they also influence the physiology of present-day humans. One such example is the Denisovan version of the gene EPAS1. This variation of the Denisovan variation of this gene confers an advantage for survival at high altitude, and you can find it among present-day Tibetans. So this gene facilitates adaptations to high altitudes and a low oxygen environment. And because the Tibetans have been living for many generations at the Tibetan Plateau, so which is high altitude, it's like 3.5 kilometers above sea level. Therefore, the oxygen concentration is very low. And because the oxygen concentration is very low, it also creates a strong physiological stress for humans who are living there. So it's important to acclimatize to this low oxygen environment. And one way that is happening in humans is to increase the hemoglobin concentration. However, if you have high hemoglobin concentration, it also causes complications and can cause cardiovascular diseases. Interestingly, Tibetans do not have such a high elevated hemoglobin concentrations. Therefore, they can adapt much better much safer at this area with low oxygen. And it's kind of associated with these variants that have been discovered in this EPAS1 gene. So interestingly, like I said before, there's groups of contemporary humans that have very high concentration of these Denisovan genes. Like you have Papuans and indigenous Australians, they have 1 to 5% of these Denisovan genes, but they do not have this Denisovan EPAS1 variant. And that's probably because there was no evolutionary pressure on these people to keep this gene because they're not living high in the mountains. Other examples are of Neanderthal and Denisovan genes that are Neanderthal and Denisovan genes that affect our immune response to different types of infections. So it's thought that these old hominins like Neanderthal and Denisovans who lived in Europe and Western Asia for more than 200 years before we arrived, that they were probably much better adapted to this environment and the local pathogens which they encountered. So therefore, we can assume that when the modern humans entered Europe and Western Asia, when they interbred with the local hominins, that there would be some exchange of DNA and genes, also the ones that are responsible for immune system. So for example, the toll-like receptors are key components of the innate immunity, and they provide a first line of immune defense against bacteria, fungi, and parasites. And they found that a cluster of 3 toll-like receptors in modern humans are derived from Neanderthal and Denisovans. So it has been shown, and it's on top of that, it has been shown that to have these kind of variants from Neanderthal and Denisovans in our genome, that it gives you a substantial advantage in immune response.
It sounds really interesting. So what might be the questions that remain unanswered?
So one important question is what makes us uniquely human. So we know that Homo sapiens are characterized by its unique capacity to create complex culture, advance innovation, figurative art, also the ability to cross open water and to spread to all the parts of the planet. We also know that Neanderthals also lived in groups and had big brains, and they also utilized tools, but these tools never developed so much over hundreds of thousands of years. They also never crossed open water and spread to all parts of the planet. So there should be some kind of difference on the genomic level between Homo sapiens and Neanderthals that could explain these features which are specifically human. And now researchers are trying to discover these kind of differences.
Okay, so my last question is, what do you think about this award?
So the Nobel Committee evaluates the nominations and awards scientists who have made most important discoveries for the benefit of humankind. And I think that unraveling the genomic sequence of extinct hominins has been a real tour de force. It was very, very challenging. And the information that we obtained is so interesting because now we get a more complete answers to important questions like who are we, where do we come from, what makes us human. So I think this is really incredible work, and it's definitely a benefit for humankind.
Okay, so this is the end of my questions. Thank you so much for your effort and nice explanations. I hope audience will learn a lot from this. Thanks.
Thank you.